Hello and welcome back to another Teach Me in 10. Today we're joined by Fredrik Edfors, who's an assistant professor in systems biology at KTH Royal Institute of Technology. He will address the gap between discovery proteomics and clinical application, discussing how targeted mass spectrometry, stable isotope standards, and rigorous quality control enable robust, reproducible data for clinical interpretability.
Hello, my name is Fredrik Edfors. I'm an assistant professor here at KTH in Stockholm, and today I will talk to you about targeted proteomics and how we can bridge discovery plasma proteomics to clinically actionable protein measurements using targeted mass spectrometry. Plasma proteomics has emerged rapidly over the years, and we can now measure thousands or even 10,000 proteins from a single drop of blood. This has different implications and has driven the field towards clinical implementation, but we are not there yet. And mass spectrometry might be targeting the high abundant plasma proteins, the proteins that we measure routinely in clinics today, while deep fractionation or affinity-based methods will quantify the low abundance range, such as Olink or SomaScan. Untargeted proteomics is very useful to find novel findings in a sample. It's great for hypothesis generation. You go out and look for proteins, but in this plot down below here, you can see that you only identify and quantify a fraction of what's available in the sample of origin. Targeted proteomics, on the other hand, is targeting a specific protein panel. You have a hypothesis and you go out and you try to validate your findings and make them ready for clinical deployment. And in order to reach the end of the line, where we all want to go as researchers, is to integrate our assays into clinical routine to provide absolute concentrations with reference intervals that will help the clinicians to take decisions. So target proteomics is actually a great fit for clinical routine. We can provide concentration units. We can put them into concentration ranges per disease or different health conditions. They're also really reproducible if used correctly, and I will come back to this later in my talk. And all of this together makes them into perfect measurements that will tell the clinicians about actionability, where you can map proteins that are used in clinics today or novel markers that we can develop to establish a clear link between a protein level or a change in protein level to a diagnosis pattern. And targeted mass spec can really provide these protein measurements in clinically meaningful units performed in multiplex in a rather short time as well. So in order to truly understand the power of target proteomics, we must just quickly revisit how bottom-up proteomics is performed today. So it's very complex in the sense that you have a lot of steps leading up to the final protein quantitation, and if you make any changes or deviations within these boxes, you will ultimately change the protein quantity in the end. So it's really important to be able to normalize for changes that might occur during this experimental sample preparation process. And if I submit a sample to a laboratory in my lab or to the laboratory across the street, we will generate slightly different protein values if we don't correct for this in the right manner. The same goes for different instruments run on different days. So it's really important to link the protein quantities and the signals that we get from the mass spectrometers with an internal standard in order to deconvolute the intensity and map that back to a protein concentration. And this is where targeted proteomics comes in. It was selected Method of the Year by Nature Methods back in 2012, thanks to its precise, quantitative and sensitive nature. And target assays are used to quantify proteins based on pre-selected peptides that act as proxies for their protein targets of interest. And the method is performed in a very controlled environment, very often with spiked standards that together solves this missing link, the standardization of the complete workflow. So we spike in anchors that can be used to deconvolute the label-free signals into truly understandable protein quantities. So we can compare proteins between each other as well as proteins across days, months, or even years, that I will come back to and show by the end of this presentation. So targeted mass spectrometry then measures peptides to quantify their corresponding proteins. And one must select peptides that represent the biologically meaningful part of the protein, as well as select which ones that are analytically robust over time and that are useful for this context. And it can be done in many different ways. You can either spike a lot of peptide standards or go for a protein standard that will yield multiple peptides and might also give miscleaved peptide and peptide information downstream. But some core design choices has to be made, and aside from selecting the peptide, you must also think about the calibration curves that should be established, as well as the quality control rules that will give you the QC parameters of your run. So you know exactly how low your CV is across days and months, and if you can deliver this quantitative level with confidence. So with targeted proteomics, we use something called stable isotope standards. These are usually peptides or proteins labeled with heavy arginine and heavy lysine, thanks to the trypsin digestion motif. These standards will give a ratio, a shift in M over Z, and the intensity difference, if measured over time and integrated, will give you an area under the curve that corresponds to the relative concentration difference between your spiked standard and your target analyte. So this is a really good way to perform analytical chemistry measurements in multiplex using mass spectrometers as the readout in your sample. It also helps to confirm the identity of the peak, which is a strength compared to discovery proteomics. It also corrects for analytical variation depending on where you spike the peptide standard, and it also enables this cross-day comparability that I spoke of earlier. So peptide standards, they are great, but I think that protein standards go a little bit further. Peptide standards, they are added kind of late in the process, and they will help correct for LC-MS variation, things that will happen in the autosampler and up until a recorded signal. But they do not capture any digestion efficiency variation, which is a huge deal in targeted proteomics and proteomics in general. Protein standards or protein level standards are added before digestion. This amino acid stretch will then follow the proteins as early as possible is the goal, but they will also generate multiple peptides per protein. They will be available when trypsin digests them into smaller peptides, and any digestion kinetic effect, either within your sample, within your batch, or something happening during that day, will be captured, and you have a larger chance of compensating for these changes using a protein standard instead of a peptide standard. So in order to go for robust concentrations, the protein standards and peptide standards, they will provide greater interlab transferability if you have the standards added as early as possible. That will also increase the chances to also correct for changes that will happen in the robotic systems up front of your sample preparation. Another key innovative step for target proteomics was the introduction of addition-only workflows. And the key concept here is that you only add reagents to your sample in order to limit the analytical bias. Another innovative step where we contributed was when David in my lab pioneered the addition step of pre-vacuum drying the protein standards that we're using, so the plasma sample can be added directly to the plate, thereby limiting the daily effect of thawing stable isotope standards and spiking them into a protein sample of interest. So this also increases the throughput since they are pre-prepared in plates, and they will also help limit the technical variability since they are prepared in large batches upfront, vacuum dried and stored in the freezer. And I just want to show some examples of how the data that comes from these target proteomics assays can look like. Here in the Disease Blood Atlas resource, we have quantified proteins using the Discovery Edge platform on a stellar mass spectrometer. And you can see that we can generate absolute quantitative levels of different proteins, here exemplified with 294 samples quantified across 3 different cohorts. We get the protein level reported as picomole per microliter, thanks to the internal standard. And we also have a feature where we get multi-peptide quant from the Discover H175, where each recombinant protein fragment standard will give rise to multiple peptides. In this case, you can see how well they trace across 96 individuals, and they act as controls for each other. And you can see that the protein level is a bit different across all the individuals, but that's also due to the fact that they have different glycosylation patterns and different folds when performing the digestion. And these are things that you would never get with peptide level standards. So I just want to come back to the stability of these assays. So the use of the spiked standards can help us to transform the unstable ratios measured across days and times in label-free mass spec into spiked stable ratios, where we get a very accurate endogenous to standard quantitation in each run. And the stable isotope standards, they help not only to confirm the retention time and fragmentation similarity, they also help to correct for ion suppression, run-to-run validation, and even translation across instruments. And just as an example, we ran one sample, digested it, and ran it on an Altis triple quad about 2 years ago. Then a couple of years later, we thawed the same plasma sample and spiked it in. So not at the peptide level, but we took it from the freezer, and we ran it on the Stellar system in PRM. And you can see the almost perfect correlation here to the right, where we used complement targeting a lot of complement-associated proteins in plasma, and with an R squared of 0.99. So this does not only confirm that we can do cross-day comparability, we can also bridge samples run year 1 to samples run multiple years after. So I just want to conclude and summarize that we now have tools to move from ion signals to true concentrations and then finally arrive at some clinical decisions. And I think that discovery proteomics has a great place in plasma proteomics in the future. I just think that we have to introduce stable isotope standards much earlier in the process, preferably already in your DIA run, so you know what peptides to go for when you're quantifying and evaluating your case and control cohort in the discovery setting. And I hope that the standardization of the technology, where you further keep on improving the addition-only workflows, will help us to standardize target proteomics one step closer to actually be able to bridge it to precision medicine efforts. And I think the biomarkers also require better measurements in order to fully succeed. And in order to come up with new diagnostic tools for the future, we have to get our act together and really work on standardization as a community. And with that, I just want to acknowledge some key persons to the work that I presented here: David, Andreas, Matthias, Jacob, and also Tasso, and the whole Edfors lab. And thank you for listening.
Huge thank you to Fredrik for joining us today. To learn more about targeted proteomics, visit the resources in the description. And don't forget to like and subscribe to stay up to date with our latest releases. Bye for now.